use the shades. You can close the shades.
All right, cool. All right, how do you want you guys, we're gonna continue on with the special sensors. Okay, awesome, well, that's way better, thank you. I just wanted to share something. My weekend I did a ropes course with the kids. I helped out with the chaperone with the church youth group, and we had all these kids, and we did the ropes course. Have you guys ever done that? You go up in the trees, climb up in the trees, and do the tight ropes, and the Tarzan where you're like on one cable, and like going from like rope to rope. Everybody did really good, it was fun. And then I told you my cat like eats all kinds of items. She's like a total predator. And I come home. So then I come home. I just had to show you this. This is kind of gross. But this is what was. That's the top of lizard, and that's his tail. So she just came in my room. She just crunches the pieces she likes, and she leaves the pieces she doesn't as a gift. So that was a happy gift to me. I just showed the kids, and they're like, oh my god, you're leaving alone. And it's so funny because then my son's eating his Halloween candy. The two of them are just crunching. My cat is crunching on the lizard, and my son's like eating his Halloween candy. It's just like, this whole place is just, something's wrong here. Okay, so what did we talk about last week on Friday? We ended with, what did we discuss? We talked about the autonomic nervous system, right? Anna, what are some of those things? You remember? Yes? Okay. Okay, the parasympathetic division, which is what? Okay. Okay, and the sympathetic. Now at this point, you guys should all know some major differences between these ones, right? How they look, their anatomy. Coming through the blinds. Their anatomy, differences between the neurotransmitters that they release, the preganglionic and postganglionic fibers, right? Some are like kind of short preganglionic fiber, and others have a long preganglionic fiber, a shorter post one. The neurotransmitters they release, and the functions that they have. So their overall anatomy, where they come off the spinal cord in the brain, right? One is more craniosacral, and the other one's more thoracolumbar. And then what are the two different neurotransmitters that they release out of their organ, their effector organs?
Epinephrine, norepinephrine.
Yep, which one?
Norepinephrine is,
Yep, and then what is the parasympathetic? ACH, acetylcholine. A, capital C, small h, acetylcholine, okay? Those are the major neurotransmitters. And then you should know the functions of what these do, right? Overall anatomy, overall structure, with respect to where do the nerves come off from the brain and spinal cord, what are the neurotransmitters that they rely on? What is a major organ or gland of the sympathetic nervous system? And I said that it makes its whole specialization is to make lots of these hormones and pump them into the blood. And it's almost like a postganglionic set of neurons that like takes on its own form in the embryonic development of it. But it's an important gland of the sympathetic nervous system. Your adrenals, your adrenal glands. Specifically, what part of the adrenals make a lot of norepinephrine and also epinephrine? And these function as hormones in your body. The adrenal medulla. Yes. Good. Okay, so really important. And cortisol, we'll talk about that later in the endocrine system, but cortisol is actually made from your adrenal glands. You've got the medulla and the cortex. So you've got the medulla in the center, cortex on the outside. Cortex has specialized tissue cells that actually pump out a lot of cortisol. And that's another major stress hormone. We'll talk more about that later. So I also talked about the special senses a little bit, sensory receptors, the different types of sensory receptors, and then we got back into the eyeball, where we left off before. You guys have now dissected an eyeball, know the major parts. So I'm gonna go through some of this before we go on to the ear, and then we get into the heart, cardiovascular system. So that's where we're going right now. Yes, make sure. And in lab today, we're gonna dissect hearts. Yeah, it's fun. It's fun though. Okay, so just to recap some of the structures of the eye, if I go back, not to the lizard, if I go back to the conjunctiva, that's that thin membrane that goes tucked underneath the palpebra and the eyelids and goes and covers the whole front of the cornea and the iris, right? So it's a transparent mucous membrane, so you can see through it. Cornea here, again, it's clear, so it allows light to penetrate through and go through the pupil, the center of the iris. We've got eyebrows, we've got eyelashes. Why do we have eyebrows and eyelashes? Some of us have more than others. That's one reason, keep the sweat from getting your eyes, keep water, sweat, fluid, bacteria, dust, germs, right? All those, you'll know that if you're ever somewhere there's all kinds of stuff blowing around, like your eyelashes and your eyebrows are a good protective mechanism against that. Also, you know if you've ever shaved your eyebrows or plucked them too much, you know that like drops will go straight in your eye. So that's a really good point. They're all protective hairs and mechanisms to help keep the eye surface from getting scratched, damaged, infected. Conjunctivitis is actually pink eye, right? So that's streptococcal bacteria getting in and invading the conjunctiva and making it inflamed and red and making more mucus, and we call it eye boogers, mocos. That's what my kids would call it when their eyes would get all stuck together. They had pink eye when they were real little. Okay, skip the lizard, sorry about that. All right, lacrimal gland, lacrimal duct, nasolacrimal duct that drains into the nose. So you got the lacrimal sac, that's kind of the outer palpebrae edge, right? So it's out on the outer part of your eye, underneath, inside of your upper sort of lid or arch of your lid. And then you've got the lacrimal sac on this end that picks up the tear fluid, the fluid production, and recycles it. And then ultimately through the nasolacrimal duct, it gets emptied into the nasal passages. In this case, this is the inferior meatus of your nasal cavity. You've got the inferior, middle, and superior meatuses that line those nasal concha bones, right, that make up your nasal cavity, outer lateral edge of your nasal cavity. These are all the membranes inside of there, so they drain. And remember I said, bless you, remember I said that saline and tear production goes into the eye, right, and keeps the eyes lubricated and fluid surrounded so they don't dry out. You gotta take good care of your eyes, and your body tries to do that this way. When you cry, you have excess production of that, right? So your limbic system is involved, and your emotional drives are involved, crying, tears. And what happens, some people get real ugly cry, right? They start snotting and stuff. A lot of people think this is a normal thing. Why? Because that excess is gets picked up and like dumped into your nose. So your nose and your whole face starts leaking. It's normal. Yes. Questions about the ugly crying?
It doesn't have to do with crying. So when you're eating something spicy and your nose starts to drip, is that like the same thing but like with your saliva?
Not the same thing, because it's not affecting specifically the lacrimal gland and making tears, although if you eat something real spicy, you will even start crying. It irritates that, makes more production. Basically, the more fluid you have here, you're draining out any toxicants, right? You're getting rid of anything that's potentially poisonous. So it triggers effects like that in your mucous membranes and in your tear glands, your lacrimal gland, to make excess fluid. You're kind of trying to dilute it out. Does that make sense? But really those are coming through your mouth and up through your pharynx, because they're all connected in the back, and then to your nasal passages. So that's a more direct route coming through the mouth and up through the nose. Yeah, and actually, cool thing about hot stuff, I like hot stuff, but you have to be very careful. There's capsaicin is a chemical in it. Capsaicin is actually a chemical that activates your pain receptors and your heat receptors, your thermoreceptors at the same time. So that's why when you eat something really spicy, sometimes you feel pain and hot, that heat that you feel. It's actually coming from a chemical that's in peppers to prevent animals from eating it. And then we eat it. And actually canning one time, I got assigned the crappiest job of everybody. My friends were like, let's do Martha Stewart canning. You wanna do the jalapenos, whatever, skinning and cutting and de-seeding? I'm like, sure. So that was my job. I got to de-seed. And the capsaicin, that causes the crying, the snotting, and all that stuff when you eat. Same effects on your hands, and it's lipid soluble capsaicin, that pepper hot component. It goes right through your skin, and it activates all those pain receptors. My hands were on fire for like 48 hours. It was horrible. I was pregnant too. I was like 8 months pregnant. I had to do research and be like, I hope this doesn't get through the placenta and to the baby. She's burning right now inside of me because my whole life. I was pissed at my friends. So I'm like, why the hell you assigned me the de-seeding role with the capsaicin? I had to put my hands in milk, in yogurt. Oh yeah, that was awful. So it triggers release of fluid, pain activation, and thermoreceptor activation. It activates all kinds of receptors. Yeah, Vito.
So like am I enough a mouse gonna cause damage? Because I know they say like, you know, sometimes don't let it touch the skin, wear gloves and stuff, like all that.
Yes, I should have done all that. I should have done all that, like, and the funny thing is, I was a grad student, I was getting my doctorate, I was like close to being a doctor. And I did that, and I came to work, they're like, what's wrong with your hands? I just played around with capsaicin seeds for, 4 hours yesterday. They're like, you didn't wear gloves? Nope. Didn't think that. Didn't think of it. Didn't know. I know if I eat it, it does that to my face, but I just wasn't realizing. And it is used, it causes heat sensation. So it's used in all kinds of different ways. It's a pretty cool thing. But this is why you get a leaky nose when you cry, right? Also for other reasons when you eat capsaicin chili peppers. Okay. Extrinsic eye muscles, I'm not gonna spend time on these. You actually dissected these. Some of you guys did a really good job of cutting around the muscles, the flaps of muscle. They attach directly to the surface of the eyeball, to the sclera. Very strange, but it's a weird, the muscles are not to bone, they originate on the eyeball sclera, and then they insert on the sclera, and they start in the orbit, or the bones inside of the orbit of the face, okay? So they go from inside the orbit to out on the sclera of the eyeball. And there's the lateral medial rectus, the recti, the superior and inferior rectus, and the superior and inferior oblique, okay? So you should know where those are, and then we've actually looked at them in dissection. I showed you this already before, how they attach, right? Superior, inferior, medial, lateral, superior oblique, it goes through the trochlea, goes like a little pulley, and then the inferior oblique that's on the bottom. Okay, this is what they do, and I talked about this already, right? And actually, these are the cranial nerves that control those different actions, those motor actions of the eyeballs. And your eyes are important, right? So we have reflexive responses of your eyeballs that you can't control. And then you have the muscles, these external muscles, the extrinsic eye muscles that you can control, right? So you can look intentionally where you need to look. When you hear something, you can look in that direction. So all these moves eyes laterally, medially, elevates eye, turns it medially, so up and cross-eyed, depresses eye, looks down, cross-eyed, elevates eye and turns it out, depresses eye, turns it laterally. So all these different motions are accomplished by these 6 extrinsic eye muscles, and the nerves that control them: abducens, oculomotor, and trochlear, okay? So you have a big area of your brain and your cranial nerves that's devoted to what you see and controlling how you see it. Your eyes allowing you to see everything you do. A lot of components of the eye, you guys already dissected. There's an anterior pole, posterior pole, external walls composed of 3 tunics. Now remember, so tunics, a lot of people, you've heard of tunics called like sweaters or tunics, right? So you layer like ponchos and sweaters, those are like tunics. The eyeball is same thing, as we call them, they're layers or tunics. And your eyeball is in these sort of layers or tunics that are the outer to the inner, as well as the inner cavities that have fluids. So the layers of the eye, and as you dissect the eye, you were cutting through those outer and then those inner layers. Those are tunics. The fluids in the eye are humors. Not like they're humorous and funny, but humors in the eye mean fluid. And there's a juicy fluid that's more like liquid out of the front, and in the back part of the eye, behind the lens, that whole 3/4 of the eye back here, there is a gelatinous one. And remember that one? You guys are all kind of timid to get it out. You're like, how hard are you going to go? I said, just dump it out, right? Squeeze it, pull it out, and it just comes out like a bloop, like Jell-O. What was that one called? Vitreous humor, right? Vitreous humor is thicker. The mass in that gelatin component to it pushes up against the back of the eyeball and keeps the retina attached. So if that gets damaged or leaks out or you don't make it, the retina can easily just detach from the back of the eye. If that happens, you have retinal detachment and blindness. And sometimes it can be surgically fixed. They can go in and tack it back up, and sometimes they can't. Okay. So the external layer has 2 regions. The sclera is the white part, the cornea is the clear part. You already know these. The limbus is the junction between those 2. So right where the 2, and you guys actually had you kind of cut on this limbus to cut out the cornea of the cow eye, okay? And then you've got a venous sinus. It's basically this venous area that allows the aqueous humor to be drained out, and then new humor is made. So it's constantly circulating and making new fluids inside of the eye, keeping it all nice and oxygenated and fresh. Lens, cornea, sclera, okay? Vitreous humor, aqueous humor floating all surrounding the lens. Behind the lens is the posterior segment, all of this, and it's all filled with vitreous humor. There's the retina back, sclera in the back, optic nerve, you all saw this, you dissected this. Blind spot and optic disc, one and the same. One is a physiological description, one is an anatomical description. Blind spot, you literally have a blind spot. And we'll do this in physiology where you cover one eye and we move something in front of you until you lose it, you can't see it anymore, it's because it goes right in your blind spot. So we need two eyeballs that have slightly different fields of view that overlap, so you never see your blind spot, but we can test it by covering one eye. Blind spot is the optic disc. The anatomy of it is a disc, and you can kind of see it. You guys saw it on your cow eyes where you kind of move the retina aside and it gathered up into that one part where you can kind of pull it, it's all attached right there. That's because those ganglion, those axons of those ganglion cells that make up the back of the retina, gather and they go into the optic nerve. So you literally have no photoreceptors, you have no rods, no cones, right there at the optic disc. You've got the middle coat, or tunic, of the eyeball. I should have said tunic, but yeah, tunic. Choroid, ciliary body, and iris. These are made of muscle, smooth muscle. Choroid, this is a lot of capillaries, blood vessels, and pigment. So it makes up that pigmented back of the eye. This makes up the muscular front of the eye, that's behind the lens. And the iris is the muscular little contraption that's pretty and colored, and it's right in front of your lens. It controls the pupil size. You looked at all these structures. Choroid, vascular, that means it's got capillaries, got blood vessels. Darkly pigmented membrane forms the back of the vascular tunic, brownish color in us, because there's melanocytes. It's brownish black in us. But in the cow, remember the cow, their choroid's a little weird. What does it look like in the cow? Yeah, it's like an abalone shell, exactly. It's like green, teal, blue, it's all shiny. It's kind of like a seashell. It has that, like a nacreous, like in an abalone shell, right? When you see the inside, it's real beautiful, that's how it looks. That helps to reflect light and help to see at low light conditions. So more light reflection, gathering those photons so they can see in low light. It kind of affects acuity, how accurately they can see, but they can see in low light. Now we're not nocturnal. We're not meant to be out dwelling at night and hunting at night, doing all those things. We have pretty crappy vision when it comes to that. So our choroid doesn't have that beautiful flash just like an abalone. It's just pretty much a lot of melanocytes and capillaries. Prevents scattering of light rays, and it actually works in that way because we don't want to scatter and reflect all that light. We want to allow the photons to come in and get centered to the back of the eye so that we can see with pretty good acuity, with pretty good light out, and not that far away. So we have good acuity, but not low light and not long range. Okay. Ciliary body, you guys all looked at this. This is this thickened ring of tissue and it circles the back of the lens. It goes all the way around. And it actually has these little suspensory ligaments, right? Little teeny little tendons, like little ligaments that attach all the way around, more like ligaments, not tendons, but attaching all the way around to the ciliary body that holds the lens in place. And you all had to do this. You flip the front anterior pole of your eye over and you try to pull out the cough drop, the lens, right? And you have to kind of cut along and kind of just separate with your blunt probe. You're separating the ciliary body and its suspensory ligaments from the lens so you can pull the lens out. It's not that hard to do though, right? It comes out. Ciliary muscle itself is part of the ciliary body, and it has ciliary processes, okay? And it has a zonule. They call it the ciliary zonule. I hate that name. I don't know why. I just call them suspensory ligaments. A lot of texts do as well. I like the names whenever possible that describe what that structure does, because that really helps me. Oh yeah, suspensory ligament. What do? Ligaments that encircle the lens and suspend the lens. Suspensory ligaments suspend the lens. They keep it in place, right? So use that one, it'll help you remember it better. Attach around the entire circumference around the lens. Oops, sorry. Suspends the lens, allow it to change shape when focusing on near and far objects. Okay, so in physio, those of you that have already had physio, you might already know this. Where's my people? You guys all had physio. Do you remember what? Behind me. And they're avoiding me right now. They're like, nope, I do not remember anything, because that was, you guys remember what this is in physio accommodation? Okay. That's okay. And online is, you probably missed a whole lot online. Take a guess. Yeah, time.
Does it change to what the need?
Yes. This is where the shape of the lens changed, exactly, to control for the need of what you're viewing, right? So, I'll give you an example. If you're looking at something right in front of you, is that the same as your lens bending light at the same way that it would if you were viewing something in the back of the classroom? No, it's very different. So your lens can either, and it's hard to tell in the cow eyes because they're all preserved and fixed, and so they're kind of firm, but really lenses are pliable. You can kind of squish them. And actually when they're clear, you can write on a piece of paper and hold it over the writing and magnify the writing. It's pretty amazing. You can take, let me take your eyeball, and now I can use it as a magnifying glass. No, maybe I shouldn't share this video. I said I complete wacko. But you can take the lens and it's pliable, right? So the suspensory ligaments are attached to the lens. If I'm doing something right in front of my face, the lens has to round up, fatten up, and bulge so that the light rays coming in get refracted or bent and it hit the back of my retina. That's where the rods and cones are. That's where I need to see the image clearly, right? Now, if I'm doing something far away, like where you guys are, my lens has to skinny up and flatten out so that the rays don't bend as much, so they go further back and they hit the back of the retina. It's all accommodating the light, AKA everything you see, to the back of your retina. Now, you've noticed old people, I'm sure you guys have seen your parents do this, your grandparents do this. When they're looking at something close, what do? They go, well, I have reading glasses, but they go like this, right? You see them in the grocery store, poor little people, I feel like, just let me read what's on their, they don't have glasses, and it's like all the way out here, because the eyeball, the lens, can no longer accommodate and bulge up like that. It's not as pliable. It loses its elastic fibers, its elasticity. So the eyeball itself, so even if you have great vision, like some old people have great vision, they can see far away, but that accommodation, it gets further and further from your face. So I have you guys do tests and physio. Most people, you guys can all have your cell phones like dead in front of your face. Like, I can't do that anymore. It's blurry, can't see it. So I have to like move it out so I'm more comfortable. And I realize I'm turning into that, where I'm in the store and I'm like, oh shit. So I have my reading glasses, so I can put them on real quick. I'm like, oh good, it's like 3 times as big now, perfect. But I have 15-20 vision, so my acuity is great. It's like a hawk from far away. But all of this comes down to the lens being able to adjust and change shape, that ciliary body pulling on the suspensory ligaments and flattening it, or relaxing and making it less taut and making it relax and bulge up. As you get older, it's a natural process of losing the lens elasticity. So you have problems with accommodation, and there's little tests we can even we take a little ruler and how when you lose that focus of what you're looking at, it gets further and further now. So it's funny, I see my kids, even like Pee-wee, he's on TikTok, and it's like right in front of his face. I'm like, how are you doing that? First of all, it's not healthy, but this is right in front of your face. You're blasting blue light, rays to your eyeballs and just directly in your face. Yeah, Gabby.
So the lens is made of elastin? What is the lens for?
No, the lens is made up of all kinds of stuff. It's like epithelial tissue.
Oh.
So it's epithelial squamous cells mixed with different types of fibers. Elastic fibers, so that part of it is that. But it's a clear, living tissue that has to be oxygenated and taken care of, and it just gets harder and more firm. It's like the skin loses collagen and elastin, those break down with aging. Sort of the same thing in the lens of the eyeball. Okay, so there it is again, lens, nice living tissue. Ciliary body is this whole structure here. The ciliary muscles in the back part, the suspensory ligaments are right here, or ciliary zonules, but I call them suspensory ligaments, and they're attached to the lens and go all the way around. So this is what allows that changing of shape, right? Really bulging up and getting rounder when you're looking at something right in front of your face. Okay. Keep going. All right, so we have iris, right? A lot of genes code for its color. People think, you have the blue-eye gene, or the brown-eye gene, or the green gene. There's all kinds of combinations, and multiple genes actually code for eye color. And some colors are super pretty. They're like hazily, honey, like mixed with all kinds of stuff. It's more than just a monochromatic or a monogenic trait. There's a lot of genes that lead to that. It's attached to the ciliary body from the front. So here's ciliary body, here's iris. What is the center of the iris? The aperture of the iris? Center? Pupil, right? And that varies in size according to light, emotional state, fight or flight, all those different things. Smooth muscle, okay? So you can't control your iris. It's not skeletal muscle, it's smooth muscle, you have no control. Pupil, round central opening. All right, sphincter, there's a couple different muscles. The sphincter pupillae. The sphincter is to constrict, contract, okay? Dilate or pupillae is to relax and kind of opens it up. So these two can work in tandem to change the size of the pupil, either making it small, constricting, or dilating the pupil. And this is affected by light as well. So reflexive response to light. Okay. Pupillary light reflex, this is a protective response. So the pupil constricts when you shine bright light in your eye. And I'll have you guys do this in physiology too, in lab. You will actually do little light, we have the little light pins. You shine bright light, you can just see it just close, just constrict way up. All right. Retina, inner layer of the eyeball. So the inner tunic, this is the deepest tunic. Superficial in the eye would be sclera, right? Superficial tunic is sclera, right? Deep tunic is retina. Now we're inside the eyeball. There's two layers, a pigmented layer and a neural layer. The neural layer is exactly what it sounds like. That's where the neural photoreceptors are. This is where the neurons are located that allow you to see. If you have damage to these neurons, this neural tissue, you are not able to see in the right way, okay? We have photoreceptors here, we have bipolar cells, and we have ganglion cells. We've got some other things, amacrine cells and all kinds of fancy helpers, but these are the main types of neural cells that are in your retina. If you have problems to any of these, you have problems with vision, okay? What are the photoreceptors? Remember I said special senses, the sensory receptors for the special senses are complete little neurons. They're like weird little mini neurons. They fire action potentials in response to a particular type of energy. In this case, the energy is light. So these are mini little neurons that respond to wavelengths of light. And what are the two main types that make up your sensory receptors and your photoreceptors in the back of your eye? What are the two main types?
Rods and cones.
Rods and cones, perfect. Rods, and they look just like this. They're named for how they look. One looks like ice cream cones and one looks like rods. Like straight, like square, and some are pointed, okay? And these basically, if I have to break it down and make it simple to understand if possible, the cones, color vision. That's easy, right? Color, see, color, cones. That's actually your high acuity, that means your accuracy, how clearly you can make out detail. Cones are your accuracy, your acuity, and your color vision. Rods then would be what?
Black and white.
Shape.
Not so much shape, no. Not so much depth, that's more binocular vision, there are two eyes with overlapping views.
That'd be clarity, like
Saturation, not so much, what else?
like clarity, like how clear you can see, or is that?
That's where the cones, and actually you could say, that's actually 3 C's, you could do clarity, color, and not acuity, but there's a C in acuity, but yeah, that's what the cones do. Rods is low light. These are like your black and white photoreceptors. These are your low light when there's not a lot of light. It's like your black and white, right? You can't make out a lot of color, but they see good in low light conditions. So you're going out at dusk or at dawn, and you're driving, and you see a stop sign. Sometimes it's really late and they don't have reflectors on it or whatever. You can't really make out that it's red, but you can see it very clearly. It's STOP, there it is. Your rods are operating at that point. So these are, I like to, it's not super accurate, it's very generalized, but these are like your black and white, your low light vision receptors. These are your color, your accuracy, what did you say, Derek, was the term used?
Clarity.
Clarity, that's good. Yeah, that's like acuity, clarity, how clearly you can see things. That's all cones, okay? So these are the 3 types of neurons that are back in the neural layer of the retina. All right, we'll keep going. This is what they look like. So interestingly enough, on the bottom here, this is the pathway of light coming into the back of the eyeball. This is the deepest tunic, okay? Deepest tunic of the back of the eyeball. And you dissected this, but it should basically just look like a little epithelial sheet that you just like ripped off, okay? But this is all the business that's in there, and this is what allows you to see, so it's super important. The light is actually would be coming through here, so the rest of the eyeball is like this, okay? And the lens would be over here. Yes? Okay. Pathway light comes all the way through. Pathway light goes through the conjunctiva, through the cornea, right? Through the aqueous humor, through the pupil, through the lens, so it's bending. Every time it goes through a different medium of liquid or solid, it bends. The lens's main obligation is to bend the light and get it to hit the back of the retina, which is right here. Now notice, a lot of people think, well, the rods and cones, okay, those are the sensory receptors. Those are the vision receptors. You've got your rods, which look like rods, and you've got your cones, which look like little ice cream cones, all back there. Now notice, they're not front and center, facing against the vitreous humor and out here. They're tucked behind 2-3 layers of other types of neurons. The bipolar cells, which are bipolar in nature, they have a cell body right in the middle, and then the ganglion cells, which have a cell body, and then they leave, their axons leave and gather up and go back to the optic disc and the optic nerve. The rods and cones are actually the furthest back away from the path of light. So photons of light, particles of light are coming in all the time, unless it's completely black, right? Coming in, and you have to traverse through ganglion cells, bipolar cells, some other horizontal and amacrine cells, and then ultimately they get to the rods and the cones. These are the ones that actually respond to those incoming photons of light, hitting them, and they activate and they send action potentials, and they send that information down the optic nerve into your brain. Ultimately, your occipital, your visual cortex, and your occipital lobe of your brain. Pigmented layer of the retina is here, where there's melanocytes and all kinds of cells, but this whole structure right here is in the back of the retina. Now this is what it looks like if we look under the microscope. Here's the choroid, the pigmented layer, vascular, has brownish pigment, okay? And then we have the outer segments of the rods and cones are all right here. So these are all right here. And then nuclei of the rods and cones, so their entire shape goes all the way down to here. So we're just looking at that piece, and then that piece, and then now the bipolar cells, and then the ganglion cells, the axons exit. Now, let me ask you why. If light has to go all the way through to strike those, to actually activate those rods and cones, why are they way back there? Why don't we flip this? Why aren't they out here? Because that's where light is first coming and hitting. Particles of light actually have to go through all those layers of cells. Yeah.
Without the other layers, it might be damaging to the rods and cones.
That's exactly it, deal. Yep, perfect. They might bake them, burn them, fry them out. They're so sensitive. That much light and particles of light hitting back in the back of the retina could actually damage the rods and cones so that they cannot, if they are irreparably damaged, they usually recycle, they make an outer segment over and over. They'll keep making new ones, but if their nuclei, if their cell body is damaged, that's it, they don't regenerate. That's it, blindness. So these are very, very sensitive and they're very, very important. So they're tucked away. So the photons of light will still make it through. They're just very, very sensitive and they're safe. They're protected in that area. And actually, it is one of those things. If you get blasted with tons of UV light, tons of photons are coming into your eyeball, that's so much for them, they will actually start to degenerate and die. Like this is why you can't look directly into UV light like the sun. If you go out and you stare at the sun, you know, where there's an eclipse, we all come up with those little gadgets so you can see the eclipse and it makes a little shadow and a cute little shape, but you can't stare right at it because it will destroy, it will bake those rods and cones. Leads to blindness, blind spots in different areas of your eyes where your vision is impaired in some way. All right, so I want you to remember that. That's the back of the eye. You have the pigmented, you have the choroid coat, the pigmented layer, and then you have the retinal layer. But the rods and cones are actually the furthest away from the incoming light. That is for safety, your anatomy of your eyes protecting the most important cells of vision, and that's your rods and cones. Okay. There we go. Okay, optic nerve. I talked about this pathway of light coming in. Retina, here's the optic nerve, optic disc, and blind spot right here. There are no rods and cones right there. So rods and cones are out in all the other area of your retina, all back here, but not right there. That's the ganglion cells of the ganglion cell axons that are gathering up and leaving and exiting. So there's no rods and cones. The rods and cones and everything are all out here. Okay, so you can't see in that location. There's a blind spot. All right, you should have all looked at these under the microscope in lab. Yes. Okay. So, and if you didn't just take pictures of them and store them away and not look at them, then you won't know what these are. But if you actually took time to take apart the